everyone. In this PowerPoint, I'm going to tell you a few tips on finding a job in different pathways post-graduation, and we'll spend most of our time describing microarrays, which are a great method. They're also sometimes referred to as DNA chips or gene expression profiling, and the majority of this PowerPoint will be very helpful with the 416 assignment. Okay, so before we get there, I want to talk a little bit about Okay, so now on to microarrays. So the big picture question when we're thinking about microarrays is trying to understand and learn which genes are turned on. Another way of saying that is which genes are expressed at a transcriptional level in which cells. And so oftentimes you're comparing two kinds of cells. So on this slide, we can see a myocyte at the top in red and a lymphocyte in purple below it. And we can see that although these two different cells have the same DNA, right? They both have the myosin gene and the immunoglobulin gene. However, they're not expressing the same RNAs. So in the myocyte, you see myosin RNA is actively transcribed, but there's not immunoglobulin RNA. And conversely, in the lymphocyte, it's the other way around. The myosin gene is not turned on, there's no RNA, but there is immunoglobulin mRNA. And the idea here is just to remind you that, cells don't express all the same genes because they have different functions. So of course, it makes sense. They'll have differential gene expression. Okay, so what do we know about trying to answer this question about which genes are being turned on at a transcriptional level? Well, we've talked about northern blots a couple of times in class, so I'm just reminding you, I drew a really very simple cartoon on the bottom right where you see a gel, and in this case, the control, you see a band, and the experimental you don't. So that would tell us that the probe is able to bind to RNA that's expressed in the control cells, but that the experimental cells do not have that RNA expressed. So there's no band. Okay, so the band is showing where the probe for this one gene is binding. So northern blots are great for giving us this information about whether an RNA is expressed or not, but we can only get that information for one gene at a time. For each northern blot, you're using a single probe, right? So you can do multiple northern blots to get information about multiple genes, but it's not, it's really not realistic or practical to ask about thousands of genes or all the genes in the genome. You just can't do that many northern blots. And so instead, people now can use a method called microarray analysis. So what is shown on the bottom left in panel B is the myocyte and the lymphocyte, and the RNA is collected from these two different cell types. It's then converted into fluorescent cDNA. It's added to the solid DNA microarray, which is usually a glass slide, and then that slide is scanned, and software is used to tell you whether or not those, which genes are expressed in each of those different cell types. And that'll become more clear as we go through the slides today. Okay, so again, what is a microarray and what is it used for? It does have multiple names. A DNA microarray is also known as a DNA chip. And what it can be used for is to simultaneously measure the transcription level of every gene in a cell. So with microarrays, you can detect changes in transcription levels for every gene in the genome for a given situation. And we'll look at a couple of examples as we go through the slides. All right, so let's first take a look and see how are microarrays made and used. So this is an image that comes from Davidson College, and they actually have their own robot to make DNA chips. And so this robot is going to, what the researchers are first going to do, take a step back, is amplify however many number of genes that they want to assay. And they're going to amplify those genes by PCR to get the DNA for those genes. So if you wanted to ask about genes in the yeast genome, and you wanted to ask about transcription levels for all of those yeast genes, that would be about 6,200 different genes. So that means you'd be doing 6,200 different PCR reactions. And then the robot actually is the method by which those different PCR products are spotted onto a glass slide. So on the next slide, I'll show you how it does that. The robot is much more efficient and precise than a human could be, and it can spot 6,200 PCR products onto 144 slides in about 12 hours. So it still takes quite a long time. Obviously, it's a lot of different PCR products. This shows you the robot arm and those little metal pins that are coming down, extending towards the bottom of the slide. Those pins are dipped into the DNA, and then that's used to contact the glass slide. So that's how you get a very small amount of DNA onto the glass slide. Okay, so what happens is that DNA will be denatured after it's put on the slide, so then it becomes single-stranded, and then it will also be covalently linked to the slide, so it'll be stable. And so you can think of it as almost like 6,200 blots, right? On that single glass slide, you're going to have 6,200 different genes spotted there. Then you're going to have a probe. And the probe is going to be a mix of cells grown in two different conditions in our example, this first example. You're always making a comparison with a microarray experiment. So in the video, you'll see a particular example, then that would be helpful to think about more specifically. And in one of the experiments I did with BIO546 a few years ago, we compared how are genes expressed in yeast in a control yeast cell comparing to yeast exposed to heat stress. Okay, so those were our two different conditions. We grew the cells in those two different conditions, then we collected the RNA out of the cells, and then converted them into cDNA. And we converted the cDNAs from the control cells using a green colored dye, and we converted the RNA from the heat stress cells using a red colored dye. So then you have these two populations of cDNAs, green and red. You mix them together, and we're going to add that mix of different cDNAs. We're going to incubate it on the DNA chip. And you'll see that in that first video that I asked you to watch. The colored cDNAs will bind to one strand of the PCR product that represents that gene, and you let that incubation go overnight. The chip is then put into a scanner that can detect the dyes, that it can detect the red or green fluorescence, and will record the locations of the fluorescence and the intensities. And that'll take about 20 minutes because there's quite a lot of data, right, to scan. The data will be stored and analyzed in a computer, and then merged images are generated. So yellow would indicate genes or open reading frames that are transcribed in both of the different kinds of cells, right, in both transcriptomes. Okay, so we'll take a pause. I mentioned a video that was listed on the assignment for microarrays, but here is the link to that video that I've been suggesting. Goes right along with these steps number 1 through 8, and it's an experiment that compares gene expression in yeast under two conditions. It's not heat stress, it's anoxia, but it's the exact same idea. So just comparing how are genes turned on or turned off when you have yeast, in two different conditions, right? What changes? Okay, so that gets us to kind of like the basic analysis of the microarray experiment, but there's actually quite a bit more of quantitative analysis that happens. So the data are going to be converted from colored spots, red, green, or yellow, to numbers that kind of give the intensity. So not all reds are going to be the same, right? A gene could be at high levels in your experimental cells, but it could be like at really high levels, so it could be like a dark red, or it could be just at like moderate levels and maybe be like a light red color, okay? So there's lots of different intensities, of course, for these different colors. And then after that, the computer will convert the data one more time to a color scale to represent the ratios of red to green. And this is how the data are presented when we read our research papers. So just as a reminder, general rules for color scale: cDNA from the control population of cells is usually green. The cDNA from the experimental cells is usually red. That's the typical situation. And then you're going to put the numerical value for the red dye in the numerator and the green value in the denominator when you are calculating ratios, okay? So let's take a look and see what these data actually look like. So on the next slide, I'm going to show you data from past BIO546 molecular genetics students. So I'm not teaching that lab this semester, but some years I do. And as I mentioned, we did an experiment comparing how gene expression changes in yeast cells after heat stress. So in the top panel, I'm showing you how the cDNA from a normal or a control cell hybridized, and that would be green because that's our control. Okay, so the cDNAs were converted, right, using a green fluorescence. And then on the bottom is showing how the RNA or cDNA from heat shock cells hybridize. So that would be the red. And then we would overlay those, right, those two different scans to see what those ratios would be. So we would have that comparison of heat stress compared to the normal, right? Okay, so we're going to zoom in on those green areas, the little green dashed boxes. And so on the far right, I have a zoomed in view of that. So you can really see how many dots there are on these different slides. It's a lot of different dots. And remember, each dot is a different gene. And if we zoom in even further within this box to the red dashed boxes, we're going to see 3 different spots or genes in the top. And we look on the bottom and we'll see that one of those dots is kind of missing or much reduced level, where the yellow arrow is pointing. So most of those dots, because they would be the same, would be yellow, right? They'd be red and green. They'd be expressed in both kinds of cells, so you'd get a yellow color. But a few of them would be different, and that lower right dot would be a green color because it's expressed only in the control cells. Okay, so that's just an example of some CMU data, I thought would be fun to share. And now we're going to move on to the paper by Stout. So this paper talks about the molecular diagnosis of hematologic cancers, and basically it's helping us understand how microarrays have helped improve diagnosis. So first we need to know what is a hematologic cancer, and these are cancers of the blood-forming organs. So this could be leukemia, lymphoma, multiple myeloma. They arise due to mutations in the immature blood cells. And instead of developing normally, the cell is going to replicate over and over, as that's what happens in cancer. So there's this overproliferation of abnormal blood cells. So leukemia is going to refer to situations where the cells that are affected are circulating in the blood and bone marrow. Lymphoma are cells that aggregate into masses and form solid tumors. And then myeloma is a tumor specifically of the bone marrow, and a subset of the white blood cells are affected. Okay, so what do we need to know about how microarray software helps find patterns to improve diagnosis, right? How is this going to work? So before we look at some actual data, we need to just understand 2 different ways that software can identify patterns in data. So if we take a look in panel A first, what we're looking at is sort of the raw data. There are 14 different patients. Their tumors were biopsied, right? The RNA was extracted, and the whole microarray experiment was done. And 29 different genes are shown here. So they are running along the y-axis, right? And then the tumor specimens are running along the x-axis. And if we just look at that initial figure, we don't see any patterns. But after a process called hierarchical clustering, you can see that the order of the genes has changed, the order of the tumors has changed, and now we see some real patterns. Especially for the 3 patients on the far right, we can see that they are very similar to one another in terms of their gene expression. All of the genes that are now in the bottom half of the figure are expressed at high levels. We see that red quadrant. And then all of the genes in the top that are now green in those 3 patients, right? Those genes are expressed at low levels. So that pattern and figuring out that those 3 patients have very similar types of tumors was only revealed because of microarray analysis and using the software with this hierarchical clustering. So that's really valuable to understand that those 3 patients are very similar to each other and that they're different from all the other patients in terms of their gene expression. That may have implications for how they should be treated, for example. Okay, and at the bottom, there's something called supervised pattern recognition. So now we're looking at just 2 of the different genes, gene A and gene B, and there are many, many more tumor biopsy specimens running along the x-axis. And at the top of the two genes, it says there's a survival plot with low survival at the left and increasing survival at the right. So if we first focus on the left-hand slide, where I just showed you a red oval, we can see gene A is at relatively low levels because it's mostly green in those samples. Gene B is at relatively high levels. There's more red. And this is correlated with poor survival. Okay? And this can be contrasted with the other end of the spectrum, where patients have good survival, gene A is highly expressed and gene B is at lower levels. Okay? So not only could treatment be impacted, but there's actually very clear effects for prognosis depending on how genes are expressed in these different patients. So that's pretty exciting to be able to figure that out. Okay, let's look at data. So this is figure 3A from the Stout Review, and this is titled Examples of Molecularly and Clinically Distinct Subgroups of Lymphoma. Okay, so what they set out to do was to look at a whole bunch of patients who had been diagnosed with diffuse large B-cell lymphoma. And that's what it says at the top of your slide. Okay, you can see some histology. It doesn't mean too much to me, but that's some histology of what that would look like, right? So these patients were initially all diagnosed with the same type of cancer. But after this microarray experiment, what we're going to see is that they've now can be categorized into three different groups. In orange, there's the germinal center B-cell-like group. Then in purple in the middle is type 3. And then in blue is the activated B-cell-like group. And what you can see is that each of the groups has a different pattern for these two groups of genes. So I've kind of called them the black group at the top and the purple group. So in germinal center B-cell-like patients, the black genes, right, that group of genes at the top that I have the black, I don't know what that's called, the black symbol, right, to as a grouping, those genes are at high levels, and the purple genes are at low levels. In contrast, for type 3 patients, all of the genes are at relatively low levels. In activated B-cell-like patients on the far right, the black genes are at low levels and the purple genes are at high levels. So the opposite of the orange patient group. So this tells us that there are distinct subsets of genes that are upregulated or downregulated in these different types of diffuse large B-cell lymphoma. Moreover, if we look at the survival curves, this is called a Kaplan-Meier survival curve at the bottom, you can see that the overall survival is not all the same. So if we split it out and we look at survival of patients whose genes correspond to germinal center B-cell-like lymphoma, you can see with that orange curve that the overall survival is much better than it is for the other two groups. Okay, so the 10-year survival is 50% for germinal center B-cell-like, and it's only around 25 or 30% for type 3 or activated B-cell-like. Okay, so overall, the authors of this paper were able to determine that when people are diagnosed, it's important to actually make a very precise diagnosis because the prognosis can vary quite a lot, and there are implications for treatment. Okay, so if we know that individuals with the activated B-cell-like lymphoma, where the survival is not as good, have the purple genes upregulated, maybe we could target one of those genes and try to decrease its expression by RNAi or something like that, right? So there are definitely possibly treatment implications as well as these diagnosis implications. This is the actual figure from the published paper. So the prior figure was from the review article, which was a bit of a summary. And I just wanted to show you how big these microarrays can be. This paper was published in 2000, and you can see the title of the paper is Distinct Types of Diffuse Large B-cell Lymphoma Identified by Gene Expression Profiling. Okay, so the review was summarizing this primary research article. And I just wanted to draw your attention to the fact that they actually looked at 96 samples, 96 different tumors. Those are along the top with that dendrogram, and there are so many of them, they're showing that dendrogram along the left as well with the labels, because you wouldn't be able to read them if they were at the top. So 96 different tumor samples, and they looked at so many different genes, it was a total of 1.8 million measurements of gene expression. 1.8 million measurements. Just imagine like 1.8 million northern blots, right? You can't even imagine that kind of number. So this has really revolutionized the ability to look at transcript levels across the entire genome. And I should say that for those of you who have heard of a technique called RNA-seq or RNA sequencing, it accomplishes much of the same kinds of types of data analysis or types of research questions as microarrays. So if you've been thinking, oh, this sounds a lot like RNA-seq, it really is. Okay. And the reason I mention that is because I wanted to explain microarray experiments because they could make sense for you and some of your experiments you want to propose in part 6 of your research paper. Okay. And if you are familiar with alternative or even more current techniques like RNA-seq, that would be fine as well. Okay, so why would a microarray experiment be useful for accurate diagnosis? If you have a disease where diagnosis is difficult because multiple diseases present similarly, you could propose doing a microarray with some control samples and then your different patients and see where patient samples where your patient sample clusters, right? If there's disease 1 and disease 2, and you can see what their gene expression profiles are, then you could take your patient and see if the patient's profile matches disease 1 or disease 2. Alternatively, for diagnosis, if you can figure out which genes are up or down regulated in a particular set of patients, those could become targets for diagnostics. Right? You could then just test expression levels of those particular mRNAs in patients. Okay, secondarily, microarray experiments could be useful to better understand the function of proteins or gene action. That all means the same, right? The function of genes and the function of proteins, we're really talking about the same thing. So if you do a microarray, you're going to learn which genes are upregulated and which genes are downregulated in patients with mutations in a particular gene. So the data that you get may inform how that gene X regulates downstream genes, right? These are going to be linked situations, right? So if there's a loss of function in CFTR, for example, and now all of a sudden I identify a whole set of genes that gets upregulated when CFTR has a mutation. Well, that's really interesting, and that tells me CFTR usually regulates those genes. And without CFTR normally functioning, those genes get dysregulated, right? They're at higher levels than they should be. So that suggests they might function in a pathway. And lastly, microarrays could be useful to develop new treatments. I kind of mentioned this earlier, but after you learn which genes are up or down regulated, you could propose therapies based on trying to alter the expression levels of those genes. So as I mentioned, for a gene that was for genes that are upregulated, you could propose using RNAi to try to decrease levels of those genes. If you're doing those kinds of proposal ideas, keep in mind that you want to use cell culture or animal models. You don't want to be proposing anything too dramatic directly in humans, but you can propose using human samples, like tumor samples, in your microarray, right? If you're not sure about how to go about thinking about this, definitely please see the tip sheet in the research paper folder. It will walk you through how to think about microarray experiments. And of course, email if you have questions.